Hello everyone. Let's get started. Today we're going to be talking about muscle tissue. Remember from our previous lectures that there are four types of tissue found in the human body. There's epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Today we're going to be focused on muscle tissue, and specifically three different types: skeletal muscle, which we'll talk about first; cardiac muscle, the muscle of the heart; and smooth muscle. Skeletal muscle performs 5 functions in the human body. Because of its location, in conjunction with the bony skeleton, it helps to provide protection to the internal organs that lie beneath. It supports the normal posture, the upright posture that human beings have, again in connection with the bony skeleton. Because it is attached to the bones of the skeleton, it also provides for movement of the body through movement of the joints. The skeletal muscle helps maintain body temperature. Through contraction and relaxation, it can generate heat. And it also helps to move both blood and lymphatic fluid throughout the body. Remember, blood is pumped throughout the body with the assistance of the heart. Lymphatic fluid does not have a pump associated with it. Therefore, the movement of the skeletal muscle through contraction and relaxation is the only source of movement for lymphatic fluids. Muscle tissue, like all tissue, is composed of both cells and extracellular matrix. When we look at skeletal muscle cells, they have some interesting features that make them unique from other eukaryotic cell types. First of all, muscle cells have a unique shape. They are elongated, linear cells, and therefore we don't typically refer to them as cells. Instead, we call them fibers. Muscle fibers are multinucleated. And the reason that they're multinucleated is because during fetal development, they are produced through fusion of multiple precursor cells. Muscle fibers have a specialized plasma membrane around them, and that plasma membrane has a name. We call it a sarcolemma. This term sarco, anytime you see it, refers to muscle. Muscle fibers contain a specialized organelle inside of them that you don't see in other cell types. This organelle is called the sarcoplasmic reticulum. Remember, that word reticulum means network or mesh. So similar to the endoplasmic reticulum, the sarcoplasmic reticulum looks like a big meshwork of tissue surrounding the workings inside of each muscle fiber. The sarcoplasmic reticulum has a specialized job to do in muscle fibers, and that is to store and release calcium ions in order to regulate the process of muscle contraction. When we look into a muscle fiber, the majority of the cytoplasm is full of structures called myofibrils. These are elongated linear structures, and they help to give muscle fibers their elongated shape. Each one of these myofibrils, as they're called, is made out of a bundle of linear filaments. These are both thick and thin filaments, and they are arranged into structures called sarcomeres. So inside each muscle fiber, there are many myofibrils, and each myofibril contains many individual thick and thin filaments arranged into sarcomeres. The sarcomere, in fact, is the fundamental unit of contraction inside of a muscle fiber. There are many, many sarcomeres inside each muscle fiber, and each individual sarcomere is going to do the job of contracting and relaxing. Muscle fibers also contain a specialized protein called myoglobin. Myoglobin is a distant relative to the protein called hemoglobin. They have many features in common with each other, including an association with iron and a specialized chemical group called a heme. Like hemoglobin, which is found in the blood, myoglobin can bind oxygen. Now, myoglobin is not found in the blood. Myoglobin is only found in muscle cells and only certain muscle cells, the skeletal muscle cells and cardiac muscle cells. Myoglobin is critical to the normal functioning of muscle because it stores oxygen inside each cell, and it allows, therefore, for ATP production to continue even when oxygen supplies might otherwise dwindle. So, for example, when a human being is in a situation where breathing is paused, myoglobin can release stored oxygen into the muscle fiber, and that oxygen can then be used in cellular respiration to help make ATP. Myoglobin is actually used medically as an indicator of a problem with the muscle. Myoglobin, again, is not normally detected in the blood, so when myoglobin is found in blood, it's an indicator that muscle tissue is breaking down. There is a disease process called rhabdomyolysis, which is a systematic breakdown of muscle tissue. Rhabdomyolysis can be detected because of increased levels of myoglobin in the blood. Myoglobin is actually quite toxic to the kidneys, and rhabdomyolysis is a medical emergency. On this slide, we're looking at both a drawing of a muscle fiber and an image taken with a light microscope of several muscle fibers, skeletal muscle fibers, lying side by side. Let's start at the top. You're looking at just a portion of this particular muscle fiber. The artist has cut off the ends and cut away some of the sarcolemma, the plasma membrane that surrounds each muscle fiber. The first thing you notice about this unique cell is that it's striped. The term that we use is striated to describe this striping pattern along the length of this elongated fiber. The next thing you notice is that this cell is packed full of these rod-like structures. These are myofibrils. Myofibrils are also striated or striped, and it's their striations that produce the overall striping pattern on the cell. If you were to take out one of these myofibrils and look inside, you would find bundles of thick and thin filaments, all arranged in a very particular pattern. These patterns are sarcomeres. They are the functional units of contraction inside the myofibrils and therefore inside the muscle fiber. Now, down at the bottom, this image taken with a light microscope shows us several muscle fibers lined up side by side. You can see where one muscle fiber ends and the next begins because of the white space that has occurred between them. Now, you normally wouldn't see this space inside of a living tissue. This is created when the tissue is placed onto the slide. It's what we call an artifact. Now, when you look at these pink stained muscle fibers, one of the things that jumps out at you is that there are many, many nuclei. These are multinucleated cells. The other thing, of course, that jumps out at you is that these cells don't have a normal shape. They are just elongated linear structures. And each one of them has several nuclei inside. Now, the other thing you can see, especially when you enlarge the image, is that there's a striping pattern inside of these cells. These stripes, or striations, as we call them, are created through the very careful, ordered distribution of the filaments inside of each myofibril inside of each muscle fiber. The filaments, the thick and thin filaments, are what create the sarcomeres, and the sarcomeres create the striping pattern. In addition to the internal workings of each of the muscle fibers, there are other anatomical features of skeletal muscle that we need to be familiar with. When we think about this term muscle, we think about what is grossly visible to us. For example, when we talk about the biceps brachii muscle, that is the muscle that helps flex and extend the elbow joint. What we call a muscle is actually a combination of many, many thousands of individual muscle fibers that are organized into a hierarchy of levels of tissue. Each of the levels in this hierarchy has anatomical structures associated with it. For example, each muscle, like the biceps brachii muscle, is wrapped in a connective tissue covering called epimysium. Remember, epi means around or surrounding. Now, if we were to take off the epimysium, that muscle would actually be composed of several individual bundles called fascicles. Now, if we took one fascicle, it too would have a fibrous covering around it. Now, that covering is called a perimysium. If you were to take off the perimysium, you would see that each fascicle contains many individual cells, many individual muscle fibers. If you were to take out one of those muscle fibers, it would also have a fibrous covering around it, and that would be called an endomysium. Now, if we took off the endomysium around a singular muscle fiber, we would have the muscle cell itself with its plasma membrane, called again a sarcolemma, and inside there would be cytoplasm, there would be several nuclei and all of the other organelles, but most of the cytoplasm would be packed with these structures called myofibrils. If you took a single myofibril and looked at it, it would be made out of a bundle of filaments, thick and thin filaments. And these filaments are very carefully arranged to overlap each other linearly, creating these sarcomeres. And again, it's the sarcomeres that form the striping pattern that you can actually see from the outside of a muscle cell or a muscle fiber. This drawing helps us to visualize this hierarchy, these levels of organization within what we call a muscle. So if you look over on the left-hand side first, you can see what we think of as a muscle, and this muscle happens to have a tendon attached to it at one end. That's very common because, of course, tendons connect muscle to bone. Now, there is a fibrous covering over this grossly visible muscle. We haven't talked about this. This is what we think of as fascia. Don't worry about that term. But just know that if I took this muscle, again, maybe it's the biceps brachii muscle, I could find a covering, a connective tissue covering surrounding it. Not part of the fascia, but absolutely covering the entirety of this muscle. That would be the epimysium. Now, if I took the epimysium off, what I would find is that this muscle is actually made out of several of these bundles that we call fascicles. Now, in between the fascicles is where I would find blood vessels and nerves, the other structures that we know run through muscle tissue. Muscle happens to be a highly vascular and highly innervated tissue. So one of these bundles would be called a fascicle, and it too would have a connective tissue covering over it. We call that covering a perimysium. Now, if I were to take one of these fascicles, and they're showing us this here, and I were to take off the perimysium, what I would find is that the fascicle is made out of many, many, many muscle cells or muscle fibers. Each one of those fibers, too, is surrounded by a connective tissue covering, and that's called endomysium. Endo meaning within. Now, if I took the endomysium off from around a muscle fiber, I would just find a muscle cell. It would have a sarcolemma around it. It would be full of its organelles, and it would be also full of multiple nuclei. That's what they're showing us in purple. And lots and lots and lots of these elongated structures called myofibrils. Now, if I took one myofibril out, what I would find is that inside are thick and thin filaments arranged in a very orderly pattern, creating these units of contraction that we call sarcomeres. Here's another drawing of this hierarchy, levels of organization that we find in muscle tissue. So again, if we start over on the left, we can see a drawing of a person's arm. This is the biceps brachii muscle, and there are some other muscles drawn into the picture. Again, this is what we think of as a muscle. And this muscle has tendon attached to it because in order to attach skeletal muscle to bone, you have to have a tendon involved. So even though we call this a muscle, this is actually a combination of hundreds of thousands, if not millions, of muscle cells. What would be inside of this grossly visible muscle, and what's not labeled on this diagram, is bundles that we would call fascicles. And if we were to open up one fascicle, we would find bundles of muscle cells. And remember, we call muscle cells muscle fibers. So this is a combination of several muscle fibers. We don't call them cells. And the reason we don't call them cells is because they look very unusual. They are elongated, linear structures. Each one of these is a cell, and we call it a muscle fiber. It's multinucleated, It has a very distinct striping pattern. And if I were to take one of these muscle fibers, these muscle cells, and blow it up, that's what we're looking at right here, I would find that most of the cytoplasm is just packed with myofibrils. And these myofibrils are simply combinations, bundles, of thick and thin filaments. And these thick and thin filaments are arranged in a very unique pattern that creates a sarcomere. So down here on the bottom, the brown structure represents thick filaments, the blue represents thin filaments. And from one line here, one vertical line here, to one vertical line here, that's a sarcomere. So from here to here, this is a sarcomere. And you can see that within these myofibrils, there's just one sarcomere after another down the length of the myofibril. And if I were to look in cross-section here, across the myofibril, you can see that this is a three-dimensional structure of sarcomeres. The sarcomere is the unit, the fundamental unit of contraction. That means that each and every sarcomere is capable of contracting and relaxing. And as each sarcomere contracts in connection with all the other sarcomeres, the entire myofibril gets shorter. And as each myofibril gets shorter, the entire muscle cell or muscle fiber gets shorter. And as all of these muscle fibers get shorter, the entire fascicle gets shorter. And as all of the fascicles get shorter, the entire muscle gets shorter. So that's why we call this the fundamental unit of contraction, because it all begins at the sarcomere.